Good morning to all of you. So today we will start with the same experiment which was done by Hershey and Chase. And it goes like this. I was telling you that the two radioisotopes which were used, those were S35, isotope of sulfur, and radioisotope of phosphorus. This was used, and the bacteriophages, they appear like this. Okay, the bacteriophages are like this. They have genetic material here in the form of DNA. So these bacteriophages, they attack these are the viruses. These bacteriophages are basically viruses. These are viruses, and these viruses, they attack bacteria. So this is a bacteria which is attacked by the virus. So this is how they do, and this is how they attack the bacteria. Now S35 and P32, these are the two radioisotopes which were used during the experiment, and the bacteriophages were these bacteriophages which attack the bacteria. These bacteriophages, some of them were cultured in this medium, some of them were cultured in this medium. Suppose if we bacteriophage with P32, I'm using the green color with P32, and I'm using the black color with S35. Now beta, as you know that sulfur is a component of protein. Sulfur is a component of protein. So when these bacteriophages are cultured in a medium having S35, it will label the protein part. And which is the protein part of the bacteriophage? This is the protein part of bacteriophage. Only this part. Only this part is the protein part of bacteriophage. That means with S35, only this part will be labeled. And when we grow the bacteriophage in a medium with P32, what for P32 phosphorus is an important component of DNA. You know, DNA has phosphorus in it. That means phosphorus is an important component of DNA. So if the bacteriophages are cultured in a medium having phosphorus or P32, which part of the bacteriophage will get labeled? It is only this part, the DNA part. This will get labeled, not the outer protein part, because protein part will get labeled with S35 as sulfur is a component of protein, whereas phosphorus is a component of DNA. So the genetic material part, which is the DNA here, will get labeled. So now after culture, the bacteriophages which we are going to obtain will be like this. The bacteriophage which we obtain here will be like this, and its DNA will be normal, and the bacteriophage which we obtain here, it will also be the normal with normal DNA. Suppose I make the normal with red color. This is normal with red color. Then the DNA, in this case only the DNA will be labeled. So for labeling the DNA, I'm using the red color. I'm using the green color to show that the DNA is labeled with the P32 radioisotope. Now, when they infect the bacteria, the bacteria has been infected by them, bacteria has been infected by them. How do they infect? When they infect, this part goes in, the genetic material goes in. So in the first case, the genetic material which goes in, it's not labeled. It's not labeled and it goes in. But in the second case, the genetic material which goes in is labeled with P32. The genetic material in this case which goes in is labeled with P32. Okay, so this is labeled with P32 which has gone in the bacteria in the bacteria, this is labeled with P32, whereas in this case it is the normal DNA which has gone in. This is not labeled with I have made just one. There are 3 4, okay, which attach to the body of the bacteria, and then they inject their DNA into them. So here I have made just again 3, but this I have shown only one. But more than one, many of them, they attach to the surface of the bacteria and they inject their genetic material in. Now after this, what happens? I told you the 3 processes which takes place is, number 1 is infection, blending, and centrifugation. Infection takes place. This infection has taken place. The bacteria got infected by this. Then blending, and the third step which takes place is centrifugation. Then I told you that this, after infection, when the genetic material goes in, then it's blend in a mixture. In a blender, it's blend, and after blending, the what we get is taken in a centrifuge tube. So this is one centrifuge tube we have obtained here. This is another centrifuge tube we have obtained here. Now the centrifuge tube, it will have 2 components. It will have a sediment part which is down, heavier, okay, and the lighter supernatant part. The lighter supernatant part, again, it will have a heavier part which is this, I think, yes, this, and I'm showing it with black color only, and the lighter supernatant part. Always the supernatant part is lighter, and in this case, the supernatant part, it consists of the bacteriophages. So the supernatant part has bacteriophages, whereas the sediment part, it has the bacterial cells. The sediment part has the bacterial cells along with DNA. Bacterial cell have DNA in it along with DNA. And the supernatant part has the protein cover, the bacteriophages. This DNA is of bacteriophage. That means this complete thing is in the sediment part. Now here the supernatant is bacteriophage. Bacteriophage plus bacteriophage without the DNA, because minus DNA, because the DNA, bacteriophage minus DNA, because the DNA has gone in the bacterial cell. Now when it was observed, this centrifuge was observed and this centrifuge was observed, what happened? This centrifuge, it has, in this case, what was labeled? In the first case, if you see, the labeling was of the protein code, because sulfur is a part of protein. So the whole protein code was labeled. So the supernatant part will be radioactive. The supernatant part will be radioactive. So radioactivity will be detected here. It has bacteriophage, which is labeled with S35 protein code. So in this case, this will be radioactive. Radioactivity will be detected in the supernatant part, whereas this will be non-radioactive. Non-radioactive. Okay, this will be non-radioactive. Now in this case, the DNA is labeled with radioactive phosphorus. So, and where is this DNA? DNA is now in the bacterial cell, and this forms the sediment. So this sediment, which is the heavier part, it will be radioactive. It will be detected. It was detected radioactive. Why was it detected radioactive? Because of the presence of radioactive DNA. Because of the presence of radioactive DNA. Because of the presence of radioactive DNA, this radioactivity was detected in the sediment. Where was it detected? In the sediment. Whereas the supernatant has bacteriophages. It has bacteriophages. And these bacteriophages were not labeled. These were normal. These were not labeled, not radioactive. Not radioactive. So this part was, in this part, no radioactivity was detected. So what did they mean? What did they conclude after this experiment, Hershey and Chase? Hershey and Chase, after this experiment, they concluded that DNA is the genetic material which is injected into the bacteria by the bacteriophages. Because in this case, where it was labeled, it was observed that the radioactivity was in the bacterial cells. So it was DNA which was transferred for infection. How did the bacteria got infected by bacteriophages? Bacteria or bacterial cell got infected by bacteriophages when bacteriophages injected DNA into them. When this DNA was injected into them. So this experiment proved that DNA is the genetic material. This was proved by this experiment of Hershey and Chase, that DNA is the genetic material. So children, this experiment is clear to you, or anything you want me to repeat in this experiment? Anything you want me to repeat, or is it clear? Anything you want me to repeat? So I'll repeat. Yes, beta. Is it clear? Yes, I want your response if it is clear, or you want me to repeat anything in this experiment. Yes, bache? It's clear. Paras Singh is saying yes. Others also, I want your response if you think that this is clear, or I have to repeat something in this. Yes, bache? Only one response I have got. Yes, beta? Yes, I'm not getting any response. Now it's Sukh Sahaj. So shall I repeat the experiment or not? Shall I repeat the experiment or not? You want me to repeat the experiment? Even if I get one message like this, I'll repeat the experiment. Yes, beta? But I should get a message. Yes, Vanshika Singh. Amit Katoch. Amit Katoch. Sanjolika. Navneet. Shruti Lakde. Okay. Yes, Amit Katoch. So Amit Katoch has not responded yet. Okay, so I'm getting the response that it is clear. So I'll switch over to the next topic, and the next topic is properties of genetic material, DNA versus RNA. So which one is better, whether it's DNA or it's RNA? DNA as the genetic material are of two types, DNA and RNA. Then genetic material is of two types. It can be DNA or it can be RNA. Now, out of the two, which one is considered to be better? Now, before this, I would like to go through some of the properties of genetic material. So, what are the general properties of genetic material? Properties of genetic material. To understand the properties of genetic material, we will start with the simple property, the first property, that a genetic material should be able to replicate. The first property, it should be able to replicate. That means that if it is a genetic material, it should be able to replicate. That means it should be able to form its copy. it should be able to replicate. The second property is it should be chemically and structurally stable. A genetic material should be chemically and structurally stable. Then it should show mutation. It should show mutation. It should undergo mutation. Should undergo mutation. Slight changes. Should undergo slight changes in their makeup, and those slight changes are mutations. Those slight changes are mutations, and these slight changes are important for evolution. As you know, if there are slight changes, those slight changes are important for evolution, and these mutations, they should be inheritable. They should be inherited. These slight changes should be inherited. That means should pass to progeny. They should pass to the progeny, and this is very important. These slight changes are very important for the process of evolution. If these slight changes will not be there, then the process of evolution will not take place. Then these should be able to express themselves in the form of Mendelian characters. They should follow the Mendelian characters. Mendelian characters. What are Mendelian characters? Again, the same thing. The law of dominance, the recessive feature, the dominant feature, how they express in the, how a dominant feature expresses itself in the presence of a recessive feature. All those are Mendelian characters. They should be able to express themselves in the form of those. Now, when we come to this, these features, now these are the general features of genetic material. On the basis of these genetic features, we can prove that which one is better, whether it's RNA or it's DNA, which is better. So RNA versus DNA, which one is better? Now, if we see in RNA and in DNA, this in RNA we have, let's make a column here, and here RNA, this side DNA. In RNA, there are two OH, these are functional groups, two OH groups, or two functional groups are present. Two functional groups are present. In, you know that in pentose sugar, okay, in ribose sugar, two functional groups are present, and here just one OH is present. And the presence of more functional groups makes RNA less stable and more chemically reactive. So because of this, the RNA is more reactive as compared to DNA. This is less reactive. Something which is more reactive is less stable. Something which is more reactive is less stable, and something which is less stable is less reactive is more stable. This is the feature that the RNA has and the DNA has. Besides this, this has uracil in it, presence of uracil. Presence of uracil. Uracil is formed by deamination of cytosine. When cytosine gets deaminated means removal of amino group, aminated, it forms uracil. So this uracil is formed by deamination of cytosine, whereas in place of uracil, the DNA has thymine. This thymine is very stable. This thymine, this nitrogenous base named thymine, is very stable. So presence of thymine makes the DNA more stable, whereas uracil is not stable. This is not stable. This is less stable as compared to, I'll not write not stable, I'll write less stable as compared to This is less stable as compared to DNA. So this less stability because of the presence of uracil makes RNA less stable. And at the same time, see, the formation of uracil is very easy. The cytosine gets deaminated and forms uracil. So a lot of uracil is formed, and which makes, so uracil is present and it makes it less stable. So this is one thing. And because of the high reactive nature of RNA, the mutation is faster. Mutation is faster, and mutation rate is faster. Of course, both of them, they undergo mutation. Mutation is seen in DNA as well as RNA, because mutation is a feature of both the DNA and RNA. And in that respect, you know, RNA being more reactive, the rate of mutation is higher. Rate of mutation is higher in this as compared to, and slower in this as compared to DNA. And because of this, what happens, the RNA evolves faster. Rate of evolution in RNA is more than DNA, so it evolves faster. This is another feature. Now there is one thing for which, so if you see in all the points, if you see, it is DNA which is more stable, it is DNA which is less reactive, it is DNA which evolves slower. So most of the, if we consider less reactivity, if we consider stability, it is DNA which is, you know, better than RNA if we consider stability. Now there is one thing on which DNA is dependent or on RNA. And what is that thing on which DNA is dependent or on RNA? That is protein synthesis. The process of protein synthesis, in the process of protein synthesis, what happens? There is DNA which forms messenger RNA, and this messenger RNA undergoes translation and forms protein. So for the process of protein synthesis, DNA is dependent on messenger RNA or RNA. So this is the one thing for which DNA is dependent on it. Otherwise, in all the respects, it is better than RNA, because stability is very important. Then only the hereditary characteristics will be stored. Now, on the basis of all these characteristics, when we consider all these characteristics, what do we get? We get a two-line sentence that in which we can complete that in which respect DNA is better and in which respect RNA is better. For that, we can say that being stable, DNA is very good for storing the genetic information. So DNA more stable, DNA is more stable. Because of this stability, because of the stability of DNA, it is very good. It is, you know, excellent in storing genetic informations. So genetic informations can be stored in DNA because DNA is stable and it will not change. So genetic informations will be stored as it is. Whereas if we consider RNA is less stable, and because of its less stable feature, RNA is very good in the transmission of genetic information. Transmission of genetic informations. So if we consider DNA versus RNA, in some respect RNA is better, but in most of the points DNA is considered to be better than RNA. But when we conclude, we can write that DNA being more stable is excellent in storing genetic information, whereas RNA being less stable, it is excellent, it is very good in the transmission of genetic information. So this is about the RNA versus DNA. I think You are able to understand it? Is it clear? This topic is clear, or anything you want me to repeat in this topic, so that if you are clear, I'll switch over to Michelson and Stahel. I'll switch over to the next, Michelson and Stahel. Yes, beta. Okay, Sukhsahaj, it is clear. Anybody else? Paras also says it's clear. Yes. Yes, beta. Yes. Any query in this? Okay, Tanish. Any query in this? Yes. RNA versus DNA. Any query in this? Any question? Because we are left with only 2-3 minutes, and if I start the Michelson and Stahl today, I'll not be able to finish it. So I don't want to start it just now. Just 2 minutes are left. Yes, beta. Any question in this? Any question in this? Any question, beta? What do you think? Which one is a better genetic material according to you? DNA versus RNA. Now, okay. Now tell me one thing. Okay, DNA. Tanish is saying DNA. That's right. Now, can you tell me one thing? Out of the two, DNA and RNA, which one you think was the first to be there? Yes. Which is older, DNA or it is RNA? Which came first? Yes. Which came first, DNA or RNA? Yes, beta? Yes, you'll have to tell me the reason for this. You'll have to tell me the reason which one was first and why. Yes. Which one can be considered as the first genetic material and why? Which one can be, out of the two, which one can be considered as the first genetic material and why? Is it DNA or it is RNA? DNA was the first genetic material or RNA was the first genetic material? Can you tell me with reason that it was DNA or it was RNA? Yes, anybody who can answer this question? Yes, bache? Yes, Sukhsahaj? You will have to give the reasons also. Yes, even if you are not sure, you can answer the question. Paras, you can also answer the question. I have unmuted you all. Yes. Yes. Now you can answer the question. I have unmuted you all. I have unmuted you all. You can answer my question. Which one can be considered as the first genetic material and why? Yes, Sukhsahaj? Okay, you are unable to unmute, Paras. Fine. You can answer me in text. RNA. Yes, Tanish, you are saying genetic material. How can we call this? And promoted evolution. Yes. Yes. RNA was, according to the characteristic features of both the genetic material, I think it was RNA which was the first genetic material. As I have told you that it was very reactive and its stability was less. And when it was less stable and more reactive, that means, and the rate of mutation was also higher. So reactivity more, stability less, rate of mutation high. So that means it is going to show changes. It is going to evolve. And finally, this after changes got modified into DNA. So this was RNA, which was the first genetic material. So children, this is the end of the class. Tomorrow I am going to start with Michelson and Stahel. Okay, children, stay fit, stay safe. I end my lecture here. Okay.